The iGEM campus, being part of the Western Ghats and a major research center, disposed of the biochemical waste products without any harm to its sensitive environment. This would not have been possible without an organized system for dealing with these waste products. So, we, a bunch of curious students, decided to take a journey along with the pipette tip to find out more about the system. We spoke to Ms. Nityarani NM, Technical Assistant Biology, to know more about the segregation of biowaste in iGEM biolabs.
Ma'am, so how exactly is the waste segregated in our biolab?
So in almost all research labs and biology teaching labs, we have three types of bins for segregating waste, biological waste. So in the blue bin, there will be general waste, and red bin, we will be discarding all plastic waste like petri dishes, then tubes, tips, album tubes, etc. And no soft plastics inside. Sterile pits, everything inside that red bin. In yellow bin, contaminated waste like cotton swabs, tissue papers, mask, then gel, aerosol gel, and polyacrylamide gel, everything in the yellow bin. So these are the waste segregation. So each lab, there will be some PhD or research students consigned for this proper segregation, and we will go and manage the waste. Those are properly segregated or not. And we have faculty, Poonam Thakor, she is concerned with particular bio-waste management.
Ma'am, so once these waste we are putting it and it's all segregated, like who comes, takes it? And
usually image people, that is Indian Medical Association goes eco-friendly. So they are concerned people. So they will collect the properly segregated waste only. So weekends, some Tuesdays and Saturdays, they usually come and collect the waste. If it is not properly segregated, they won't take. So we should properly segregate it, and then they will collect it and go.
Ma'am, so will they kind of check?
They will also check. So we usually check and kick, and they also will check once again, and then only take it.
The primary sorting of bio-waste is handled by the housekeeping staff of our institute. Hear from them.
കവറിൽ ലാബ് നമ്പർ എഴുതി വയ്ക്കും. പോയിട്ട് ഈ ലാബ് ഗ്യാസിൽ എന്തെങ്കിലും കംപ്ലൈൻറ്റ് ഉണ്ടെങ്കിൽ ഈ ലാബിൽ തന്നെ വരുമ്പോൾ നിങ്ങൾ തിരിഞ്ഞു കൊടുക്കണം. പിന്നെ യെല്ലോ കവറിൽ ജെല്ല് പോലത്തെ സാധനവും, പിന്നെ നിങ്ങൾ ടിഷ്യൂ പേപ്പർ ഉപയോഗിച്ചിട്ട് കെമിക്കൽ ചേർന്ന് വരുന്നില്ല, അതും മഞ്ഞ കവറിലിടണം. പിന്നെ മറ്റുള്ള റെഡ് കളർ കവറിൽ നീഡിൽ പോലത്തെ സാധനവും, പ്ലേറ്റ് പോലത്തത്, പിന്നെ ഗ്ലൗസ്, പിന്നെ അതിൽ സൂചി ഇടാൻ പാടില്ല, സൂചി പ്രത്യേകിച്ച് വെച്ചിരിക്കണം. ഒരു കവറിൽ, ഈ മഞ്ഞ കവറിലായാലും ഈ റെഡ് കളർ കവറിലായാലും സൂചി ഇടാൻ പാടില്ല, അത് മാറ്റി വെച്ചിരിക്കണം.
The segregated bio-waste in color-coded bags are off to the bio-waste processing plant in Palakkad.
Following the truck, we reached Image, the biomedical waste management plant in Palakkad, run by the Indian Medical Association.
There are 4 categories of waste: yellow cover for incinerable anatomical waste, red cover for plastic materials, blue cover for glass bottles, and white puncture-proof bags for needles and sharp metals. Strict segregation has to be followed at the source of waste generation. Each cover has a unique barcode printed on it through which waste can be tracked via GPS. First, we visited red cover waste management plant. All the red bags are autoclaved for 1 hour. There are special biomarkers added that change color on complete sterilization. The plastic is then segregated according to different types. Then it is washed thoroughly, shredded into pieces, packed into bundles, and sent to recycling company to make third-grade plastics. Even for needles and metal items, they are autoclaved, shredded, and collected in the needle pit. Later, they are transported to iron furnace to be recasted for reuse. In the case of glass bottles, they are washed with hypochlorite solution, segregated into different sizes, and later sent to recycling company. Moving on, we visited incinerator plant for combustible yellow bag waste. The waste is burnt at 2 different high temperatures, 800 degrees Celsius and 1000 degrees Celsius, and after multiple filtration, the flue is left out and ash is collected in the ash pit. Compared to the wet incinerator, which makes use of water, fully automated dry incinerator have very minimal flue, and this difference can be clearly seen in the effluent flue concentration. There were 3 incinerators, and the emission purity will be checked by OCEMS, online monitoring of industrial effluent, and the data is sent to Pollution Control Board on a daily basis. All the water that is used for washing the waste materials are treated in effluent treatment plant. The wastewater was treated through bacterial decomposition and several ultrafiltration for being reused for washing vehicles and gardening purposes. To ensure the reusing water's purity, water samples undergo quality check by chemists on daily basis. Finally, we had a short interview with our guide Mr. Naveen M, who works there as an MEP engineer.
So, the scope of our motive is to periodically collect it from the healthcare establishments, remove from there and accept it here and segregate it and dispose it in the same way within a prescribed time limit. This was a challenge faced by us and we are striving on it each and every day.
Later, we got an opportunity to meet the former president of Image, Dr. V. Vijaychandran.
What do you feel like the future of Image would be like to on forward? Like, do you want like the government to be involved in it or like build more plants?
Of course, we need, even now we need more plants. In Kerala alone we need around 5 plants. In the law itself it says that it should not be transported more than 75 to 100 kilometers away apart. When you collect it, it should be disposed or it should be treated within 100 kilometers. Above, here also we have to have more than 4 or 5 plants. But we have tried, we have by giving price we have purchased land at so many places. But people don't allow to start this plant. They are totally against this waste treatment everywhere. Some attitude in our people that waste should not be taken to their place, should be taken to some other place.
Didn't you face it in Palakkad at that place?
Palakkad is also facing this problem. Even now it is why we are facing this problem.
An organized biowaste management system relies on the effectiveness of primary sort that happens within the research labs. Being a research facility with multitudes of labs generating tons of waste each day, a student community who is well aware of the segregation procedures is quite essential. To know more about the initiatives put forth by the Institute in this regard, we interviewed Dr. Poonam Thakur, Biosafety Officer in charge for School of Biology Labs.
So ma'am, at the starting of like every semester or like 2 semesters, we are having this safety classes for all of the students. In that, as the segregation and the management part also included.
Yes, so we give a general biosafety orientation to all the new students who start working in the lab to make them familiar with how to work safely in the labs. But almost 50% emphasis is given on how to segregate the waste correctly and how to contain the waste production and what kind of waste should be handled in what kind of ways while working in the lab. So that's quite a major part of our orientation to the new students.
So in general, not like in our institute, what are like the guidelines that has to be taken care like from higher authorities also? Do we need some special kind of permission or something for like working in the...
So all biological labs and institutes throughout the country are running under several kind of rules and regulations, of course. So they are dealt in very different types of categories. For example, there is this IBSC, Institutional Biosafety Committee. So they have to ensure that all the modified DNA, RNA or any such constructs are given due approval. Their sequences are submitted in the central repository. So you cannot just start working with any biological agents by yourself. So all thorough approvals need to be met. Similarly, to start working with animals, you need approval from Institution Animal Ethics Committee. So there also the protocols for working with mouse, etc. have to be very, very clearly laid out. In the same way, waste management structures are also to be put in place before you are allowed to even, make the labs running and operational. So same way, there are rules and regulations for all steps of the way to be able to run the lab, and that's safe for all the institutes across the country.
What would you like to say to the students who are like just entering major or like just starting their work in the lab?
Only thing is enjoy science very, very responsibly. If you are careful, it will be helpful to you as well as to your colleagues around you.
When this journey filled with curiosity, memories, and a lot of new information comes to an end, we would like to thank everyone who supported us. Special thanks to Dr. Hema Francis, our Institute Medical Officer, who helped us get in touch with Indian Medical Association.